Thanks, Al. Also, I recognize that this is the last talk. I really appreciate everyone sticking through it. And I also appreciate Hal for pointing out how long I've been in the lab. I'm as old as PCTC is. I'm going to talk about not iPSCs, but a joint project with Tian Pan Lab at UCSF. And this inconvenient finding, as Hal mentioned, of how the metaplastic alveolar basal cells in a fibrotic lung can come from type 2 cells via aberrant signaling, specifically focusing on the aberrant mesenchymal signaling. There you go. As we appreciate, the most abundant anatomical feature in the normal lung is an alveolus, and the most frequent cell type we find in these alveoli are SFTPC positive type 2 cells. In the distal airways, we can also find basal cells marked by cytokeratin 17, as we see right here, an early marker of basal cells. But in the case of an fibrotic lung, IPF lung especially, the anatomy is quite distinct in the sense that there is a lot of alveolar structure, not in the least but replaced by fibrotic lesions, but also general destruction of what an alveolus should look like. And there is a general reduction of SFTPC positive type 2 cells and quite a few K17 positive or K5 positive basal cells that come up in these alveolar regions of an IPF lung. And if you look at the total lung, especially comparing between a normal lung and an IPF lung, two things happen. One, there is a remarkable loss of both type 1 and type 2 alveolar epithelial cells as a fraction of total epithelial cells. And in conjunction, there is an upregulation of basal cells, also again as a fraction of total epithelial cells, that a number of groups have made observations about over the past 5 to 6 years. And the correlated loss of type 2 cells and basal cells is something that's been plaguing us in our lab at least. And we wanted to test a simple hypothesis, is that whether these type 2 cells lose their identity and then transdifferentiate into basal cells in these fibrotic lungs. To answer these questions, we employed a number of different techniques. The first, in vitro, is what we did. In that, we first isolated type 2 cells by way of flow sorting by staining for EpChem, marks for total epithelial cells, and HT2-280, which is a specific antibody for mature type 2 cells in a normal lung. And this is single-cell RNA sequencing of these freshly sorted type 2 cells, which shows a uniform expression of canonical type 2 markers such as surfactant protein C and ABCA3, and virtually no expression of basal markers of K5, K17, or a club cell marker SCGB1A1. We took these cells and put them with a MRC5 fibroblast. These are embryonic cell type, embryonic fibroblast cell line. And this has been shown by a number of different groups to promote and maintain type 2 identity, which is what we saw. Majority of the colonies that came out of this system were type 2 colonies, at least by protein staining. However, if we switch the mesenchymal cell in this co-culture system, and now instead of co-culturing with an MRC5 cell line, we use adult human lung mesenchyme sorted from normal lung, but first cultured and then put into this co-culture system. Just as early as day 7, we see general loss of complete type 2 identity and acquisition of K5 protein staining. And by day 14, there is a complete loss of SPC protein staining and induction of basal cell marker K5. And we were curious whether this is just an artifact of how we are isolating these cells, whether that has anything to do with the fact that anytime you isolate cells out of from normal human lung, it's still an injurious event. It's a pretty traumatic event for a cell. So we wanted to see if we can culture these type 2 cells for long term and whether they can still maintain their transdifferentiation ability. To do that, we used a protocol that was published in Cell Stem Cell by Tata Group out of Duke, where we can isolate type 2 cells and then maintain them over a period of anywhere between at least 8 to 10 passages as reported by them as type 2 cells, which is exactly what we did. We isolated these cells, put them in culture for anywhere between 3 to 4 passages, almost close to 2 months, and we harvested these cells, confirmed that the majority of them are still SFTPC positive type 2 cells. And indeed, more than 96, 97% of them maintain their type 2 identity. And then we looked to these group of cells, which are all SPC positive type 2 cells, and subjected them to now the same conditions that we had just described, culturing with either MRC5 or culture AHLM. And 14 days into the culture, we saw the same results as we saw earlier, that even these purified culture type 2 cells, which effectively function like a cell line, a primary cell line, they're pure type 2 cells. And if you put them with MRC5, they maintain their type 2 identity. If you put them with AHLM, they lose their type 2 identity and acquire basal identity. So the next logical question in our head was, well, okay, this is great. This is in vitro. Does this occur in vivo as well? To do that, we performed a injury plus transplant assay that we are very well established in our lab. What we did was we took NSG mice, NOTS-gate gamma, heavily immunocompromised mice, injured them with bleomycin. 10 days into the injury, we divided all of these mice into 3 different groups. One group received just the type 2 cells. Second group received type 2 cells plus MRC5 cocktail. And the third group received type 2 cells plus AHLM, the adult human lung mesenchyme cocktail, kind of mimicking our in vitro conditions. About a week to 10 days later into this transplant, we harvested the lungs of these mice and stained for a number of different markers. What we saw was kind of interesting is that even in the group of mice that received just the type 2 cells, it was easy to find K5 positive basal cells coming out of the type 2 cells that is marked by HNA. HNA is the human nuclear antigen. And of course, we also found a number of different engrafted regions which were surfactant protein C positive type 2 cells. And that didn't really change if the mice received a cocktail of type 2 cells plus MRC5. We still found a number of basal cell regions which were HNA positive and type 2 cell regions which were also HNA positive. Again, we found something similar in the third group, type 2 cells plus AHLM, where there is a bit more appreciable number of these basal cell engrafted regions, and also there were some regions where it was just type 2 cells. However, when we quantified all of these regions, we found that in the groups of mice that received either just the type 2 cells or MRC5, 75% of all engrafted regions, all HNA positive stained for epithelial marker regions were type 2 cells, were stained for SPC and not for K5. However, in the third group, the ones that received AHLM along with the type 2 cells, only about 5% of them retained their type 2-ness. And then the 75%, maybe 74%, 73% of these regions were now completely basal cells and devoid of any alveolar markers that we could find. And so we were clearly the co-transplantation of this AHLM along with the type 2 cells promoted this basal cell switch, just kind of like in our cell culture system. So we looked at them a bit more carefully. And for that, what we did was we transplanted type 2 cells, but now with RFP labeled AHLM. And we found that there are few regions where we observed both SPC positive and K5 positives in the same parts. There is a green arrow here that shows a K5 positive cell, and red arrow that shows a K5 positive and an SPC double positive cell, with a white arrow that is just an SPC positive type 2 cells. And what we found was that in these regions where there was an active differentiation going from an SPC positive cell that was engrafted and now into a basal cell, this AHLM were right next to them. This is just a HNA staining and a merge image, which really suggested to us that at least during the active regeneration, differentiation of type 2 cells into basal cells in an injured fibrotic host, AHLM by their anatomical or geographical proximity might be playing a role. We don't really know what that role is just yet. We don't know whether these AHLMs can can sustain themselves in this injured lung for a long period of time. But at least in this active transdifferentiation, we see their juxtaposition next to these parts. So clearly, AHLM has a role to play. But what is that role, and what makes these cultured AHLM so special? To answer that question, we looked at the isolated mesenchyme from IPF lungs, so freshly sorted from IPF lung. Then we also isolated mesenchyme from a normal donor lung, never seen any culture directly off the sort. And then the third group of the cells that we looked at was the cultured AHLM that we know promotes the type 2 transdifferentiation into basal cells. And we performed single-cell RNA-seq. When we look at all of these three different cell types together, we find a number of different clusters of interest. In the fresh AHLM, the ones that never saw any sort of culturing conditions, the majority of the cells were alveolar fibroblasts, distal fibroblasts, which we know promotes and maintains a type 2 identity, which makes sense. What was interesting is that the cultured AHLM and IPF had two clusters that are specifically increased in these two mesenchymes. One was these BMP low clusters, where a number of a group of fibroblasts that expressed a number of proteins that are known to antagonize BMP signaling. Just as BMP signaling is known to promote a type 2 fate. And second, the CTHRC1 positive cluster that is recently discovered and recently identified to express a number of ECM proteins. It is high in TGF-beta and TGF-beta signaling, and is generally associated with active remodeling regions in an IPF lung. So really, this cultured AHLM, at least by this single-cell RNA-seq and by looking at these couple clusters, had increased TGF-beta signaling via CTHRC1 cluster and a low BMP signaling via this particular group of fibroblasts. So next question was that, well, if we increased TGF-beta signaling and inhibited BMP signaling, can we mimic this particular phenotype in vitro? That's exactly what we did. We co-cultured type 2 cells with MRC5, which in normal conditions, without any perturbations, should and do promote and maintain type 2 fate. 49% of the cells are SVC positive, around 3% are K5 positive. But we add BMH1, which is a pan-BMP inhibitor. We see that the number of type 2 cells have decreased. There are some double positive cells, and 35% of the cells are now basal cells. And then we add TGF-beta in this system. We find that the pure type 2 cells marked by SPC protein expression are further decreased, and now the majority of the cells are basal cells in this particular system. So which goes on to suggest that in an otherwise normal system, if we change TGF-beta or BMP signaling, the type 2 cells don't behave just as type 2 cells. So the question then was, how do these type 2 cells adapt to basal fate? Is there a switch? Is there a one-step conversion? Or is there some sort of a progression that they slowly acquire basal fate? To answer that question, we performed a time series single-cell RNA-seq experiment where we sequenced freshly sorted type 2 cells, day 0, if you will. Virtually all of them are type 2 cells. Then we performed single-cell RNA-seq analysis at day 7, 14, and 21 in this organoid system of total epithelial cells. And we observed that as early as day 7, we see quite a few basal cells, and there are some type 2 cells. And interestingly, these type 2 cells and basal cells are linked by these two cell populations that we chose to call ABI, or alveolar basal intermediate 1 and 2. And day 14 into the system, we now see emergence of club cells in vitro. And by day 21 into this culture system, we see emergence of ciliary cells as well. So we really see over a period of 21 days in vitro, we see a repertoire of all different cell types that we can find. And most of them are linked between type 2 cells into these area cells via these intermediate cells. So we're curious about what are these intermediate cells and how are they really described in fibrotic lungs, if they are. And the last couple of years, there's been much more detailed analysis of IPF lungs by no very small contributions by the IPF Cell Atlas. Especially we looked at this particular data set coming out of Krosky Lab at Vanderbilt, where they analyzed a number of fibrotic lung diseases, and we focused on the IPF out of all that. The type 2 cells, type 1 cells, and what they found were these two cell populations, which they termed as transitional AC2s, which were highly similar to type 2 cells, and these K17 positive, K5 negative basaloid cells, which they found to be persistent and almost terminally differentiated, if you will, in an IPF lungs. So we took this data set and we tossed in basal cells in this particular analysis. And we found that, well, at least by UMAP projection, the basal cells are sitting right next to these basaloid cells. And when we do the pseudotime trajectory analysis, it's a prediction to suggest that there are two ends to it anyway, at least, well, 4 ends to it. And knowing from our cell culture system, we put AC2s as time 0. We see a nice progression from type 2 cells into these patch-like cells, is how the Tata group described them, into this either type 1 cells or into the basal cells. And that was kind of exciting because when we looked at our organoid data, we saw exactly the same thing. But we saw the type 2 cells via ABI1 and 2 emerging to the basal cells, and then eventually gave rise to club insulated cells in vitro. So we thought that, well, these intermediate cells and the transitional AC2s and basaloid cells may be somewhat related. So we looked at the molecular signature of these populations from organoids and IPF lung, and we found is that type 2 cells are like type 2 cells, basal cells are like basal cells, but of interest, the transitional AC2s share a number of genes which were with the ABI1s, and the basaloid cells, or the K17 positive, K5 negative cells, share the features with ABI2s. Those are just some of the selector markers that we looked at. So that gave us a bit more confidence that what we are able to show in vitro in our pretty reductionist 2-cell organoid system has at least some similarities, I should say remarkable similarities with what's been described in IPF lungs. So we wanted to see what are these signaling pathways that govern these AC2s and then ABI1s and ABI2s, and then if there is anything that we can learn from that. So we looked at signaling pathways by way of looking at upstream regulators of these 3 cell types. And what we found was confirmatory and more assuring is that some of these alveolar niche factors like GATA4, FOXA2, NKX2.1, BMP and Wnt signaling are highly upregulated in type 2 cells and somewhat in ABI1s. However, the fibrotic niche factors such as HAS1 suggesting activation of Notch signaling, or ECM proteins such as fibronectin, collagen, and TGF-beta, another molecule that we looked at earlier, are highly upregulated in ABI2s, and they're also upregulated in the basal cells eventually. So we're pretty confident that what we're looking at here has pretty high similarities between earlier described transient populations in IPF lungs. So we went ahead and we asked, well, can we find these cells in IPF lung in situ? And this is a relatively low power.
Jimmy, sorry, 1 minute, last minute.
All right, thank you. So we found these clusters of ABI1s, ABI2s, and basal cells, all in the same pods, suggesting the linear relationship between them. And then knowing that IPF is relatively, or not really, it's pretty heterogeneous disease. It's really characterized by this somewhat seamless transition from a normal-looking alveolar structure to thickened alveolar walls into more microcystic regions. And we wanted to see what are these ABIs lying there. And we found that as we go from a normal-looking region into a thickened alveolar wall and into the microcystic and eventually into heavily bronchialized regions, we find this that the ABI1s dominate these normal-looking regions, and then eventually giving rise to ABI2s, which are marked by K17, and eventually into the basal cells. So we see that as the injury progresses, we see the progression of the ABIs into the basal cells. And last but not least, we wanted to see how are the mesenchymal cells related to these ABIs. And we did in situ for what we did earlier, SPC and K17, but now we threw in the CTHRC1, the earlier mesenchymal marker that we saw. And then we found that these ABI1s were, this is just a CTHRC1 notes, the ABI1s were marked by SPC and K17 low expression, but almost never next to CTHRC1. However, the ABI2s marked by K17 expression and negative for SPC and K5 were almost always right next to the CTHRC1 positive cells, and so were the basal cells in an actively remodeling regions. And this is a quantification of the same, which really linked the aberrant mesenchyme with these aberrant cell types that we see in the fibrotic lung. In summary, what we saw is that type 2 cells in a normal region and normal alveolar niche, by no small parts with due to the BMP signaling perhaps, are maintained as type 2 cells. But as mesenchyme and I'm sure many other signaling events in an IPF lung occur, the aberrant signaling events, maybe TGF-beta and then many other signaling pathways, cause these type 2 cells to lose their identity in this progressive pattern, and then acquire the basal identity through ABI1s and ABI2. Some of these ABI2 continue to persist into this basaloid cell, in the microcyst, but some, which we don't know how, do transdifferentiate into basal cells, and as we know, basal cells continue to
we have to close. We're all busy. 7 minutes over.
Thank you. And this is my funding, and this is any questions I can answer via emails if there is no time.